we're very lucky to have Ian Smith join us to talk to us about plant pathogens, in particular Armillaria and Phytophthora, which I'll let Ian tell you much more about.
All right, I'd just like to introduce, to start with, what are diseases, and we talk in terms of causal agents, and they can be either biotic or abiotic. And so abiotic, of course, is just the droughts, the fire, wind, hail, waterlogging, lightning, pesticides, and all those other things. And we call them disorders. Whereas the biotic ones, we've got the insects, the defoliators, the vertebrates, and those which are called, I call the pathogens, which are the fungi, the bacteria, the viruses, and phytoplasmids and nematodes. And then we have another group called parasitic plants. That can also cause issues with trees. But to describe how these things work together, we have a concept called the disease triangle, which describes the interaction of the main three factors that influence the development of disease. And we look at it like this. Let me show this. So we have the susceptible host, we have a pathogen, which has to be virulent on that host, and then we have the environment, which has got to be conducive to a compatible interaction between the host and the pathogen. Without any one of them, of course, without a susceptible host, of course, you won't get any disease. Without a virulent pathogen, you won't get disease. And without this environment, even though you might have the pathogen and a host present, without the environment that's suitable, you won't get disease either. So what I'll look at tonight, if I get time to go through it all, but I think I'll at least cover the Phytophthora and Armillaria. We'll have a look, perhaps look at some defoliators that might have an influence on some of the plants here in the Dandenong ranges. The first one I'd like to talk about is Phytophthora, and that's what my career started in the mid-1970s, working on Phytophthora cinnamomi, which I'll talk about a lot more in a minute. But the Phytophthora diseases are in the Mycota, which they're not actually in the fungi at all. They're in the kingdom of Chromista. They've recently been taken, well, recently in the last 10 years, were taken out of the fungal kingdom and put in their own kingdom called the kingdom Chromista. The Phytophthora itself is derived from the Greek. Phyto means plant and thora equals destroyer. So it contains some of the most significant plant diseases in the world, with over 125 species described worldwide, many of which are well-known root shrubs, fruits, and foliage pathogens of trees and shrubs. Probably the first main disease that was, of course, recorded of Phytophthora was the potato blight caused by Phytophthora infestans. That was the very first Phytophthora that was described back in 1864, whereas it caused disease in the 1840s on the potato crops in Ireland. And it actually caused the mass starvation and immigration of over 2 million people throughout the world. So if you've got Irish ancestors in Australia, you probably find you can blame that on Phytophthora infestans actually causing the migration to Australia. There are some interesting other stories that go with Phytophthora infestans, and there's a book that's called Magical Mushrooms, Mischievous Molds, which is a great little book to read, and it talks about how Phytophthora infestans won World War I for the Allies by causing the starvation of the Germans. So it's a long bow for pathologists, but they like to take credit for it. So we've got over 124 species now described worldwide, many of which are known on fruit ones. But the number is actually that have been found is actually increasing each year. So in Australia, we have about 32 species, and they consist of this whole list here, as you can see. You'll see cinnamomi, which is the one I'm going to talk about most. There's another new one called Phytophthora phallax, which occurs in the ash forest, and another one, multivora. So these are ones that have appeared in the last 15 years or so to cause issues. And then there are a lot of exotics to Australia that we haven't got here yet. And we probably won't have time, but I'd love to talk at some stage to talk to you about Phytophthora ramorum, which is one that's a really critical one if it gets to Australia and have a big impact on the ash forest. So Phytophthora cinnamomi is considered the most threatening environmental plant pathogen. It has a potential to also cause economic and social loss. It's microscopic in size, so you can't actually see it in the soil. It thrives in moist conditions, and it feeds on the roots and basal tissue of living plants. The root damage hinders the movement of water and nutrients, and resulting in weakened or dead plants. In a historical snapshot, it was introduced to Australia probably in infected plants by early European settlers. Circumstantial evidence suggests that they might have been responsible for epidemics in agriculture in Queensland in the 1890s. But it was actually first described in Sumatra in 1922 as the cause of cinnamon tree cankers, where it got its common name cinnamon fungus, even though now it's not a fungus. It's still often called the cinnamon fungus. It was first found in Victoria in 1935, and actually in Australia, in cricket bat willows in Melbourne, when the plantations there became infected. In native forests in Victoria, 1938, it was Dieback was reported in the coastal forests at a place called Partelli's Crossing between Nowa and Orbost. But at the time, they didn't know what it was. And again, in the 50s, reports in the Grampians and East Gippsland. And the Forest Commission pathology at the time couldn't find out what the cause was at the time. However, in 1956, Phytophthora cinnamomi was associated with the death of native heathland in New South Wales. And then the big one was in 1965 with Dr. Frank Podger, who did his thesis on jarrah forests and the death of them in Western Australia to find that, to categorically find out that the death was caused by Phytophthora cinnamomi. In 1969, just only 4 years later, it was associated with the death of silvertop ash on the lowland forests of East Gippsland and an understory in the forests of Brisbane Ranges. In the 1970s, the pathogen again was associated with Dieback at Wilson's Prom and in South Gippsland. And in 1974, oh, be quiet, it's Siri's asking me questions. 1974, Queensland, there was a devastation of avocado crops in 1974, which led to the development of new chemical control measures, which I'll talk about as well. It's widespread in horticulture and a significant issue in nurseries and gardens in particular. What do we look for for disease symptoms with Phytophthora? Because you can't see the pathogen itself, it's microscopic, you really have to look for disease symptoms to see whether your tree has it or not. It infects the fine roots of the tree, then it moves up through the fine roots into the main roots and then into the stem, causing this brown discoloration in the trunks of the tree. You can see here. You often get exudation of sap around where the lesion is on the tree. But Chyno on its own is not a definitive, because Chyno exudation often occurs through any sort of response to trees to injury. Disease symptoms, I say, reciprocal chronic drought. The leaves wilt, they turn yellow, then brown, branches die, and it can happen sometimes within a very few months of the first symptoms. How do we identify it? What we call the signs of a pathogen, they're all microscopic in size, and here you can see these are called the sporangia, the chlamydospores, and these ones here, the oospores of the pathogen. But it can all be only done down the microscope. And sometimes you'd think when you're looking down the microscope, things are waving back at you. So it's found throughout Australia, all the way from Cairns to Perth in Western Australia and in Hobart. So you can see the distribution. It's actually distributed a lot more in Victoria than it's shown in this particular map. It has a direct impact on causing significant diebacks of eucalypts and losses of understory species, primarily, say, in the coastal forest, woodlands and heathlands. And you can see this is the sort of damage you get of grass trees in the Brisbane Ranges, where you get the pathogens invading through the soil, and it's gradually killing off the grass trees as it advances down what we call an advancing front. And this is the sort of dieback here that you get in this case it was South Gippsland, but in eastern South Gippsland and the Grampians. This is the Wilson's promontory, so you can see how it's got into the soil and it's killing the plants as it goes. And again, this is the Otway Ranges. In Western Australia, you can see these quite devastating pictures in 1993. This was taken in one of the Fitzgerald and Stirling Ranges in Western Australia. You can see what it looked like there. And this is in 2010. You can see it's been completely devastated by the pathogen. But But it also has an indirect impact. As you can imagine, many of the animals that require shelter or use those particular plants for food can be quite badly affected. And some of these there's actually 22 native mammal species have more than 20% of their known distribution in diseased areas in Victoria, and 5 species are classified as either rare or endangered. So how do we look at this? How do we examine this? And as I mentioned, the disease triangle is something that we looked at in terms of how we might manage the pathogen. And so we looked at the host plants, we looked at the pathogen and the environment. And I'll go through a little bit about each to show you the sort of approach we take. First off is the pathogen. It has three spore types. It has zoospores, which are tiny little spores. These are only 10 microns in size. The sporangia are about between 20 to 60. These things here are about 20 to 60 in size. I'll just show you what they look like when they're coming out of actual sporangia. The chlamydospores are produced in the roots, and they're a resting spore, which are important in the survival and spread of the pathogen. And then there's the oospores, which in Phytophthora cinnamomi has a very rare that you actually ever find these things in nature. You can do it on an agar plate, but you can't actually find them in nature. But this actually is really important in some of the other Phytophthora in generating new hybrids, which we all heard about hybrids with COVID. So these are the sort of things, the way that these pathogens develop new hybrids themselves. And I might talk about that a little bit later. Next, this is the life cycle of the pathogen. So it's in the chlamydospore stage within the dead root systems of a tree. They germinate, producing sporangia. These spore sacs come out of the root, and then the little zoospores swim out and they attack a new healthy root. And then slowly but surely the healthy plant becomes infected. The pathogen moves up through the root system and into the trunk, and then kills the tree or any plant. We can use, sometimes in native forests, we can actually use what we call indicator plants that where you get these sort of massive deaths, it's a good indication of the fact that the Phytophthora is present. And grass trees, Anthera australis, and another small plant called Isopogon pteridophyllus, if they're dying in a site, they are a good indicator of the presence of the pathogen. But most of the way detection and isolation is done is through soil and plants and through laboratory tests. Soil baiting, for instance, where we collect soil from these disease sites, it can detect down to 1 to 2 infected propagules per gram of soil, which is a very good test. You can do it through direct plant isolation also. You can take the root systems of a diseased plant and plate it straight onto an agar plate and isolate it that way. We can also isolate it from water sources now, and I'll show you how we go about doing that in an upcoming slide. So the baiting operation, this is how we do baiting. And it's an easy thing to do if anybody wants to do a bait. They could just get you just get a plastic container. Does it show? It does show across the on there. Okay. You just get a plastic container, put about a third full of soil, flood it with water, and then you get some baits. You can either, in this case, I use Eucalyptus seberi cotyledons that I germinate specifically for this purpose, or you can use just Packham pears and put a Packham pear in there. And what happens is that this part of the cycle here is, I guess, picked up by the zoospores. Rather than infecting a root, they infect a little leaf here. And in case of Eucalyptus seberi, they actually, when they germinate, they're actually purple underneath. You can see them purple on here. And as the cells get broken by the pathogen, the anthocyanin, which is the cause of the reason the cotyledon is purple, the anthocyanin leaches out into the water, and it turns from purple to green. And so you've got actually a good little indicator of the presence of a pathogen in your bait. And same with the pear, you get this lesion on the actual side of the pear. And that's a good idea that you've actually got a Phytophthora present. And I often encourage students when I'm teaching them that if they're wanting to go in test soils, well, you can do this to start with and screen your samples before you go sending them off to the lab. And you can then, that way you've screened out the negatives, and you don't have to then send a lot of samples to the lab to see what's present. And it can save you a lot of money if you're actually having to pay for the tests. So you can see here you look down the microscope, and along the edges of these leaves here, you get more of these sporangia appearing. These are characteristics, the shape, size is characteristic of each different species of Phytophthora, and often you can quite pick up Cinnamomum, at least from this process. There are so many others present now. We really have to go to this next stage and plate onto selective agar, and then go to a PCR test. Now, we all know what PCR tests are through our COVID things. Well, same thing happens with Phytophthora and things. They can do PCR tests to see, another DNA test to see what pathogen it is. Another way you can actually sample the water by, we created these mesh bags, and we put our baits in them. And I'll show you what they look like in a stream. You float them in a stream with these, either a rhododendron leaf or a eucalypt leaf in them. Rhododendron leaves are a good leaf to use as well. And if the pathogen's present, well, let's say they change colour or they get these lesions on the leaf. This is something that we started using for quite a while. Unfortunately, there's a potential issue with other animals that live in the streams when you leave this thing for a couple of weeks in a stream. So we've stopped using this and gone to the next method, which is what we call the bottle of bait technique, or BOB. And this just relies on the use of host material again. We use either cotyledons or rhododendrons, or in fact we use both normally in a bottle. We go down to a water source like a river, and we just collect the water in the bottle and throw the baits in straight away. And then you incubate those for at least 3 days with the leaf pieces, and then again you take them out and place them out. You can also do water filtration. Just take water and filter it through filter papers and plate those filter papers, and you go from there. This has a problem in the sense that you've got to get them done on a lab in 12 hours. So I don't tend to use water filtration as much. We've developed some other new techniques where some of the new Phytophthora species, which I probably won't get a chance to talk about today, and perhaps it's time for another session, which are aerial Phytophthora species, like Phytophthora infestan, the very first potato blight. Some of these new Phytophthoras are also aerial Phytophthoras, and they're splash dispersed. And we tried using rain gauges we put out into the ash forest actually, and we isolated Phytophthora called Phytophthora phalanx from the water dripping down out of the leaves of the trees. So the trees were 60 meters high, and you're collecting these samples down the bottom. So it's a useful technique to try and see whether we've got any of these new Phytophthora present. How does it get around? Again, infection and spread. Infested streams is one way that it spreads. If there's an infected nursery somewhere near to a stream, that drainage from that nursery can run into a stream, and then from there it can be run down the streams. These little spores that come out and swimming along, and then they can get sucked up by the next person's water source as he sucks it back up to use on their nursery. So it's really important to have clean water when they're in nurseries. But a big one is in the past has been infested gravel pits, and these have been where you get infection occurring along the boundary of the forest, and the spores get washed into the actual gravel pits. The gravel pit picks them up, and the machinery picks it up and puts it in the truck, and they take it off and use it along the roads. And it's believed this was a source of infection in the Grampians, the Brisbane Rangers, and Angerhook Parks, because what you saw was that those advancing fronts were actually coming from the sites where they just put fresh gravel on the roads. They didn't know at the times. We're talking about the 60s and the early 70s. So often they didn't know that that was the source of the infection. Nursery plants is another really big one, which I can spend a long time in this because nurseries that don't have good hygiene control can actually end up spreading the pathogen long distances around the state and across Australia. And many of the, for instance, the mature trees that people grow, they're sitting on the ground, which gives opportunity for any pathogens that might be in the soil getting up through into your pots. And often they grow what they call resistant hosts, and I'll talk about the hosts in a minute. But people grow resistant hosts in these nurseries, but they're not resistant, they're tolerant. And in the pots, the pathogen can continue its life cycle quite happily on the fine root systems of the trees without actually them showing any symptoms. And this can therefore be a way that the pathogens can move around. I'd hate to be, like if you're in a land care group and you grow all these trees and then you go and plant them out on the sides of the road and it infects other trees around it. It would be, I'd feel really bad if I were in that situation. Again, mud adhering to bulldozers and was thought to be the reason why the pathogen got into the Verica range in Wilson's Promontory, and there was possibility to tracked vehicles used during training manoeuvres during World War II was also a way that it got through. The pathogen often gets picked up on soil and transmitted that way. Again, they didn't know anything about it. The firefighting one was traced back where the bulldozer came from was in South Gippsland, where we know now the pathogen was present already, and they spread it accidentally. Again, they didn't know at the time. Local spread can also be can happen on boots, and I know that Gretna Westy, who was Dr. Gretna Westy from Melbourne Uni, who did a lot of work on Phytophthora back in the 70s and 80s. She had a little path that she went from the main road down to her healthy site, and unfortunately, the track down to her site became infected from just people walking up and down there. So you've got to be really careful when you're using it. Make sure you wash your boots and things before you go in from a diseased area into a healthy area. And native and domestic animals have been suggested as being a cause, but they generally don't pick up too much mud on their shoes. I mean, things like pigs and things are known to spread the pathogen in Queensland in the mud and in the wallows that they produce, and so those sort of things can be a problem, particularly in the future now when we're getting apparently pigs are starting to get into Victoria, and it may cause some significant issues there. There were some long shots thought at one stage back in the 70s where they actually took some worms and put them into a soil that was infested with Phytophthora spores, and then they fed those worms to birds and then identified Phytophthora coming from the droppings of the birds. It's a long shot, but it's a possibility. But it's compared to a bulldozer containing a few hundred kilograms of soil on it. It's no real comparison. But even itself, the pathogen, once it's in the soil, it spreads via motile spores, the zoospores, those little tiny ones you saw come out of that sporangia. They swim through the soil. It's the phenomenon greatly aided by surface runoff and subsurface water movement downhill. It can move across slopes too and uphill via uproot-to-root contact between infected and uninfected plants. You'll remember that it can only move through soil when the soil is actually at field capacity. It can't move when there's not enough water between the soil particles. The control side of things, we can also develop a lot of these various eradicants. This is really only useful in nurseries. I use this quaternary ammonium compound, such as Phytoclean. You can purchase them from Woolworths and Coles. If you just look on the bottle for quaternary ammonium compounds, when you're wanting to use them for washing down machinery, it's much better than chlorine or sodium hypochlorite, and because the other things tend to rust your machinery, whereas the quaternary ammonium compounds don't, and they're a good product to use for washdown and hygiene. Soil sterilant such as methyl bromide is not used at all anymore because it's a greenhouse gas, and it's only ever specially used now in some quarantine procedures. There are protectants. The original Phytophthora infestans potatoes were protected using Bordeaux mixture, which is a copper product. And the newer ones out now, there's like Mancozeb Bravo and Thalamine Capthine. They are protectants. What they do is they build a barrier between stopping the Phytophthora getting into the root systems in the first place, but they're only there as long as the chemicals is actually on the plant, and as soon as it washes off, all the pathogen gets in. Whereas the systemics fungicides such as metalaxyl and phosphonate and dimethomorph are ones that you can apply, and they will get into the plant and they will be traveled through the plant through to wherever the pathogen is. The phosphonate, this phosphonate one is an interesting one. This is the one that was developed for the avocado industry back in 1974, was brought out. It's a PO3, which is closely related to, as you can imagine, to PO4, which is phosphate. Phosphonate is a product that when it's in the plant, it actually causes the plant to become more resistant. It actually works on the resistance of the plant, so building up the immunity rather than actually impacting directly on the pathogen as much. So it's an excellent chemical, and it's really very safe. It actually used to be used as a mouthwash apparently at one stage there. So it's not that things that of the past have been found to be issues now. So, but no, this is we're not recommending being used as a mouthwash, so don't do, but using it. But phosphonate is an excellent product to use if you want to build the resistance of your plant up, and particularly prior to any infection. So if we look at the disease triangle, what can we do with a host plant? So the host of Phytophthora cinnamomum, there's over 1,000 species been recorded as hosts worldwide. Chestnuts, walnuts, rhododendrons, Proteaceae, Liliaceae, Pachydermaceae, susceptible Banksias, but not all of them. Eucalypts vary considerably in susceptibility. In fact, eucalypts were in the 1970s when I started with the department. A lot of we're still online here.
Yep.
Oh, that's okay. My other laptop went off. That's all. Eucalypts. One of the very first things that the department did when they found the disease in East Gippsland was to have a look at what species is being affected, and we did a lot of what we call pot trials in our glass houses to see whether resistance is not. This is Eucalyptus seberi, and you compare infected and uninfected. You can see the difference it was, and these things eventually died. We did a lot of plantations as well. So we planted several different species from various groups to try and find out which ones might be tolerant to the pathogen. And from those trials, we found out that we had the gum, box, and the ironbarks were quite tolerant, but the stringy bark, ash, and peppermint species were quite sensitive and susceptible to it. Although it was interesting that we found that when we planted 100 like silvertop seedlings down in East Gippsland, we would lose 95 out of the 100, which only left 5%. But it was interesting that would suggest that 5% of the population might be more tolerant. And I'll come back to that potential later on. So this is the sort of symptoms they got in the various fields. It's the eucalypts, again, the Banksias, the jarrah, even pine trees can get affected by it as well. In horticultural crops, pineapples, rhododendrons, oaks, chestnuts, walnuts, almonds, avocados, they're all potentially affected by Phytophthora cinnamomum. When we look at the hosts, native host species across Victoria, you can see that the high populations of native species that are susceptible to it occur in the Grampians, the areas down towards the Brisbane Ranges here, the Otways, Wilson's Prom, East Gippsland, and there are sections up here at Kinglake as well. We look at the environment. That's what are the environmental parameters that cause it to become conducive to the interaction. And we all know what an epidemiologist is now, I'd imagine. But in terms of epidemiology for this pathogen, we've got temperature conditions here, minimum growth between 5 and 6 degrees. So if it gets below 6 degrees, it basically kills the pathogen in the soil. The optimum for growth is between 24 to 28, much the same as what most of us are like, I think. And maximum for growth is 32 to 34 degrees. And above those temperatures, it starts to kill the pathogen off in the soil. So you can see that temperature plays a big role in the pathogens in the epidemiology. Soil moisture is the other one, as I said. You have to have fully saturated conditions, as you can see here in this case, where you get these perched water tables just above the soil. You can see here the soil here's got a clay pan with sandy soil above it, and that's the ideal conditions for the pathogen being able to get into trees and plants. And another thing we found, though, that was the plants themselves have to have high plant tissue water content for spread into the collar, and we did that by looking at trees and seedlings, and where we had some that were stressed, in fact, and some that were unstressed. In fact, this particular experiment, we thought that stressed trees would be more likely to be more susceptible than unstressed trees. And so we went along expecting to have the hypothesis that these ones were going to die before these ones. But in fact, the mildly stressed trees, in this case, it was only stressed from high stocking rates. So we had seedlings that were coming up like 1,000 seedlings per hectare, whereas these ones were only in the 10 seedlings per hectare. So they were on their own. And we got tiny lesions developing on the stems of the ones that were in a competition, and really large stem lesions that actually ring bark the trees with the ones that were unstressed. So it seemed that by releasing the stress on the tree, and this tree was of course growing better, it was probably producing more sugars and those sort of things for the pathogen to use. And we seem to think that it allows the pathogen to move faster than the plant's defense mechanisms can actually pick up the fact that it's present. And so we end up with these massive lesions. And it explains why wet sites tend to favour the pathogen. We found also that soil types was important. Here we've got an organic matter red soil, crasnosome soil, in with a sandy soil next to it. And we grew, in this case, it was a Eucalyptus obliqua in either side of each other. The one on the left-hand side died with the one on the right-hand side. And so organic matter, diseases are favoured in soils with low organic matter. The organic matter seems to have lots of other bugs and things in the soil, which tends to kill off little zoospores before they get a chance to get onto the root systems. The mycorrhizae in the soil form little sheaths along the roots and stopping the pathogen getting in. So high organic matter soil is actually suppressive to the actual development of disease. And so when we look across the Victoria, at least, we can see here that we've got the red one are duplex soils, which are the ones that potentially have issues with could have issues with Phytophthora. But if we also add to that the temperature regimes, we can see that the blue here is too cold, whereas the others moderate and these areas here are too dry. So unless you actually water some of these sites, which has been known to do, sprinkler systems and things where they're growing susceptible plants, that you've actually had some substantial deaths of trees in those horticultural environments where they haven't taken account of the pathogen being present on the shallow soils. But so we've got most of the areas through here are too cold for the pathogen to be, and also have really good high organic matter soils. So when we overlay our risk with our species, we can see this is what we come up with in terms of biodiversity risk in Victoria. You can see the low risks, the orange is moderate risk, and the red patches here are high risk areas. You can see throughout Victoria where the worst risks are. So that's using the disease triangle to work out where we best target our research management procedures. And so as I say, we've a pathogen, a primary pathogen. There is variation in susceptibility both between and within eucalypt species, and I'll come back to a second with that to explain how we might use that in our disease triangle. The physiological condition of the host, water potential is important in disease development. So the trees that are not stressed at all will be more severely affected. and soil types and structure and climatic conditions, rainfall and temperature have a big impact as well. So how do we use this management, use what we know in the management of the disease? In the case of susceptible hosts, we try and improve our host resistance. We can breed for resistance, we can change the species, and we can improve the health of the plant. And this is, we can make sure that it's growing well, good nutrition, and it's, and in case of the use of phosphonate, which can actually help improve the resistance of a plant. The pathogen down here, we need to try and see whether it's mostly set on trying to eliminate or suppress it and prevent its introduction into a healthy area that's uninfested at the moment. And then, of course, modifying environments, not easily done in a native forest environment, although you can select, there is some things you can do and make sure you don't put road culverts across the roads into susceptible forests. That would help to reduce the impact of high moisture content. And you can select non-conducive environments for planting. So our disease management approach is based on the susceptibility of plant communities and the ecological and scientific importance. It's so far, the pathogen is so widely spread throughout Victoria now, it's really a case of we're trying to contain the existing outbreaks and prevent spread into uninfested areas. So there's a lot of effort going into trying to find these uninfested areas that are of high ecological value, and then take precautions to make sure you don't introduce it to those areas. And so you classify the vegetation according to risk and locate and protect the high risk areas. And monitoring is such another important thing, to monitor what's happening. Also promoting regeneration of infested sites, and I'll come back to that in a second, and promoting awareness, of course. These are great TV ads from Western Australia, but I haven't got time to show you them now. Again, mainly hygiene and quarantine is used for protection of high-risk sites, and these are done often through like washdown stations, making sure that your dozers and things are clean before being transported from one area to another, particularly from an infested area to a healthy area. Even with fire, trucks can be washed down. And this is not only just for pathogens, it's also for weeds and things to prevent them getting into areas where they're not wanted. And of course, this was the Wilson's Prom fire. They actually put a lot of effort into making sure that the people working on the firefighters were actually washing their vehicle, they're washing their boots and all their machinery before going from a diseased area to a healthy area. Fungicide application, this phosphonate, as I was talking to you about, in Western Australia is actually, and there has been some places in Victoria as well, as applied by air. They seem to be getting 5 to 6 years from one application of the fungicide control over the Phytophthora in this way, and it's a great project that's going on over there. You can also do what we call stem injections. These are really simple chemjet injectors that will inject into the trunk of the trees and help provide protection from the pathogens in the soil. Just come back to the interest-specific variation. As I say, this actually was done with Regnan seedlings, or Regnan saplings actually, and we inoculated a whole heap of them to see what sort of variation in resistance there was in the population. And you can see here, it varied from very large lesions. These are each one of these is an individual tree. This is a what they call a control, which is where where you put a plug of agar into the trunk of the tree without any Phytophthora. And this is two different. We were actually looking at two different strains of the pathogen to see whether there's any much difference in them. And in this case, you can see that the introduction of the pathogen caused these massive lesions, but it showed incredible variation in resistance across the plants, and that was another thing towards what might we do. So how can we take advantage of this resistance in our populations? So in one place in South Gippsland, we actually went around collecting seed from yellow stringy bark trees that were still surviving on some of the sites. And these were grown in nurseries and then replanted back onto the Dieback sites. And that was quite successful, but unfortunately plantings are not as quick to grow as the normal native forest would be. these forests tend to grow fairly slowly anyway. So that was one approach. The other approach was to try and make use of the natural resistance within the population. And so what we did, we actually took an area, we clear felled it and burnt it, and then we sowed it with 200,000 viable seeds per hectare. Now, if you remember rightly, I said before that between 5 and 10% of the actual trees in the plantations that we actually tried to establish on these sites, there was about a 5% resistance. But if you apply 200,000 viable seeds per hectare, you're going to end up with potentially 10,000 trees coming back up that will survive. And so we tried that back in the 70s, and we started growing these things. 5 years after treatment, we had this sort of growth, because a good ash bed is needed to get good growth of these plants. If you just put it straight onto the soil, they tend to shrivel up and die and don't survive. So a good ash bed is really good to get a good kick along in the first few years. And we had a lot of deaths over the seedlings over those 5 years, but by 25 years afterwards, we've still got a really good growing forest. And I must admit, I've got to get back down there to see what it's like now after 40 years. And I can see it from Google Earth. It still looks pretty good. So that's the sort of thing we can do for how we can approach a particular issue like Phytophthora, to look at the various things like improving host resistance, excluding the pathogen, and trying to modify environment. Current investigations in Sassafras Creek by Bill and his team, because he was concerned that Phytophthora cinnamomi might be an issue. And this is the sort of results we've got from isolating the pathogen. We can see here that this one here was actually, because I mentioned to Bill, we did get an isolate from up that way in Sassafras Creek back in 1990, and but it wasn't causing much of an issue. And in the ash forest, I say, there's generally too cold for Phytophthora to be a problem. It can become an issue if, for instance, if there's a lot of drainage down the hill during the summertime when it's warm enough, so you get this temporary introduction to pathogen, which then dies off again in the winter. But so Bill collected all these samples from out the site, and we got no result from any of them, which was great to see that Phytophthora at this stage, at least, haven't been an issue in the Sassafras Creek dieback. But as you can see here, though, we did get isolations of another pathogen called malaria, and they'll deal with that next. Unless anybody's got any questions regarding Phytophthora, I'll move on to the malaria issues.
We've got a couple of questions, Ian, that we might.
I can't see them.
Well, we've got them. The first one was from Helen about how we might ensure that we're not buying infected plants from a nursery. Is there any way that we can be sure?
It's a really important one, because what I tend to look for is ask the nursery, are they accredited with the Nurserymen's Association of Australia? No, it's an IASA approval process. Unfortunately, it's only about 10 or 20% of nurseries are, and or that they're taking precautions to prevent pathogen becoming an issue, because NIASA accreditation has a whole series of steps that the nursery has to take into account before they get accredited, and that they include your water supply is clean that you're getting in, that your plants are your soil, that your potting mix that you're using is clean, that your plants are kept up off the ground so they can't get infected from any soil that might be around, and that you're taking in hygiene precautions. So all inputs into the nursery are clean, and so that all outputs of the nursery are clean. And so that's an important point in deciding whether or not to buy your seedlings from there. So you could ask them what steps are they taking to keep Phytophthora out, and they might be able to tell you they should include all those particular aspects of it.
Thank you. Another one that's quite relevant for us is whether bush mulch might spread infection, because it's quite common for us to get bush mulch from the national park onto our work sites.
Yeah, bush mulch. It's not likely to have, unless it's got a lot of soil in it. It's not likely the mulch itself, unless the mulch is coming from diseased trees that has been chopped up. I mean, if you've got plants coming out of a garden that are have been mulched, put back in the garden, then you're quite likely to be spreading the pathogen around your garden that way. But ordinary mulch shouldn't be an issue. In fact, mulch is a good thing to have into your soil because it helps to add organic matter, and that organic matter is really important in building up the other microbes which actually can kill off the pathogen.
Yeah, great. And Karen's asked if nappy sand would knock off PC.
I'd imagine it's nappy sand's, I've forgotten the chemical now. It's hydrogen peroxide, I think. I would imagine it would. Yeah. But it's
final question there on PC is more of asking you to comment. Glyphosate herbicide is a phosphonate, and this is a controversial issue. Do you care to comment? The other question following on that is what happens with glyphosate? Does that affect PC?
Interesting. I'll try and deal with both. Glyphosate and phosphonate are not the same thing. Phosphonate is just simply PO3. It's quite a it doesn't kill plants at all. Well, unless you use a high rate and you burn it with phosphonate, you can do that if you do use a high rate. But it's only used at a rate of 5 mils per litre, and it's such a really low dose. It's not related to glyphosate at all.
Yeah.
I mean, glyphosate might have phosphorus in it, but it does a different mode of action.
Yeah.
The other one, what's it?
What's the effects of glyph on PC?
It's interesting. Glyphosate will actually potentially cause plants to become more susceptible to these things. We had a nursery back in the mid 80s where they had a significant issue. This is an open rooted nursery where they grow the seedlings in the ground and then they dig them up and then to carry them off. And they had an incredibly bad issue with Phytophthora cinnamomai in the nursery, and they couldn't work out what they were applying fungicides, and it still wasn't seem to make any difference. And then overnight, the pathogen disappeared, and we wanted to try and find out what the cause was. And it took us quite a while to work it out, but it was a change in the use of the different herbicides. They were using what they call a pre-emergent herbicide called an atrazine, a simazine, and they were using that to stop grass growth and things like that coming up through the nursery. And it was actually giving the plants like an AIDS type thing, and Phytophthora was becoming much more prevalent than it needed to be. And overnight they changed from simazine to Dacthal, and the pathogen didn't like Dacthal, and so the nursery got back into full production again. It was an amazing thing overnight. So we don't know often when we use a particular product for a certain thing, whether it's going to have side effects. I know that if I've used glyphosate in glasshouse trials, and it does promote disease in Phytophthora in seedlings. But so it's and there's a whole lot of other herbicides that are used to do the same thing. But there are also a number of herbicides that actually work like fungicides and kill them off. So always be careful when you're applying any chemical, that there's not a secondary response from that particular chemical.
And then the last question we'll just quickly finish up this section with is whether there are any plant distributional changes due to PC. Well,
you saw those areas in the Brisbane Ranges where the grass trees were just being destroyed, and what tends to happen is it goes from having a woody shrub understory to grasses and sedges filling the sites afterwards, and so that has the impact, the flow-ons to all the animals that use those particular things, not having the seed coming on the grass trees and the other trees. So yes, there has been some major changes in distribution due to Phytophthora.
I reckon that's caught us up on that, Ian.
Good. How are we going? We got time for me to continue on?
Yeah, we've said 9, so
That's all right.
we've got you for a while yet.
That's okay. That's all right. Well, you can see from that slide that I put up, you can see that there's armillaria throughout the sites too. And I'll just quickly go through what is armillaria. So this armillaria is a worldwide genus of about 40 species. We have 5 species recognised in Australia: Armillaria lutebubilina, Armillaria hinulea, Armillaria novaezelandia, Armillaria pallida, and Armillaria fumosa. But Armillaria lutebubilina in Australia is considered to be the most virulent of species. Armillaria is one of these strange things. If you actually Google it, you'll find that it's called the humongous fungus. It's actually the biggest living organism on Earth, or it's disputed to be the biggest living organism on Earth. In the US, there's a place where the pathogens started off in the middle and it's slowly moved out, such that it's they can trace back the same pathogen in the center of the circle as it goes out about 5 kilometers. And so it's considered to be the biggest living organism if you dig up all the this mycelium and put it all together. So it's an interesting story. It's a basidiomycete. It's a proper fungus. It occurs naturally, mainly in eucalyptus forest. And I need to highlight that it is a native pathogen, this one. It has damaged approximately 2,000 hectares in the mixed forests of Mount Cole and the Wombat State Forest. It is active in the Dandenongs ranges and particularly in home gardens, and attacks many species of both native and introduced trees and shrubs. And it can kill trees and shrubs of any age. Again, it kills plants like Phytophthora. It kills plants through root and collar rot. It's initially, in this case here, you've got this grey gum here. This is down beside the Puffing Billy tracks down at, or was down here in 2003. This is what this tree looked like. And a year later, it was dead completely. And you can see the fruiting bodies down the base here, which I'll get to in a second. But the foliage wilts, leaves turn brown and die. In this case, armillaria can also cause a sapwood rot. It actually rots the actual sapwood. Again, like Phytophthora and some of the others, it can weep kino from the trunk, but as I said, it's a non-specific response to a wound, but they do produce that. So how do we identify this? One of the simplest ways is to when you're not, it's not in June or July, the simplest way is to peel the bark back from the trees, and you look underneath the bark for this white mycelium growing underneath the bark, between the bark and the wood. And that's the mycelium of it's quite distinctive fan shape, creamy white mycelium sheds. Sometimes it goes a bit chocolatey brown, and many of the other armillarias produce what's called a rhizomorph, and in the soil, the mycelium actually merges together to look like a little shoestring root, and that helps it to get from one root to the next root under the ground. But this Armillaria lutebubilina very rarely, at least in Victoria, produces these rhizomers. So the easiest way to identify it is in late May to early June to look for mushrooms around the base of an affected tree. The colour is a yellow-brown, yellow-olive colour. You can see it here. This is what was on that grey gum. The cap size is 4 to 15 centimetres in size, so it's quite large. The stalks are up to 25 centimetres long. So this is 25 centimetres long. The gills are attached and spores are white. See how the gills here come in, they go straight into the stipe. That's what's called gills attached. And the spores here are white. You'll notice often when you look underneath the plants here, you'll see a white powdery on the top of the cap below the other one, and that tends to give it away. But if you really want to, you can take a cap out and put it on a piece of white paper and leave it overnight in a nice dry spot, and it'll produce a spore print. The other thing is this is to have a ring around the stipe. That's another important characteristic of armillaria. I don't promote eating it, but I was introduced to it by my mentor, Dr. Jeff Marks, by taking a bite of it, and it has a very lingering bitter taste. we actually went to a site and we found the armillaria fruiting bodies. He said, "Oh, take a taste." And I took a taste of it. I said, "Oh yeah, it tastes a bit like a mushroom." He said, "Okay, let's go." And we got back in the car, and about 2 minutes later, it hit the back of my throat, and it was just, it was so bitter and lingering that he wouldn't even stop the car to let me have a glass of water. I said, "You'll still learn." I learned not to take anything from him. There's only one thing that really can be confused with if you've got all those characteristics, is a fungus called Gymnopilus. It also has the same characteristics, but the difference is that this has orange spores, and it's very easy. All you do is put your fingers into the gills and they'll come out brown, and you know that that's a Gymnopilus and not armillaria. The other thing with Gymnopilus is generally they fruit much earlier. They tend to fruit in April rather than in late May and early June. So you'll get these things start to appear on trunks of trees in say in late April. And there are Gymnopilus is basically wood decay fungi. It doesn't tend to kill trees, but certainly can help in the decay process. As I said, it does not infect the soil like Phytophthora does. It spreads from a woody food base, a tree stump. It moves from a tree stump in, as you can see here. It moves down through into another root systems, and then affects the next tree, and then it slowly spreads further and further and further until such time as the tree dies. You'll get fruiting bodies appearing on the trunks as they die, and those spores can potentially infect another stump or another tree where there's a wound. This process is very rarely seen. It's I've yet to see this really happening, that it could be attributed to spores coming in. It's generally this part here is the cycle that you see most of the time. So again, we'll look at susceptible hosts. One thing about we don't know what hosts. I haven't ever been to find out what a resistant host of Armillaria luteburbilina. There are plenty of species lists from overseas which say this is Armillaria resistant, but that only applies to overseas species. We haven't got a host list for Armillaria luteburbilina. It seems to attack most things. Most control measures work to control the pathogen here and the environment. So what can we do? We can reduce the inoculum. This is the sort of approach that's taken in home gardens and in council gardens, is where you get the infection. You have to remove the whole trunk. That takes out most of the inoculum sources from the site. We can create physical barriers. In this case here, you have the Armillaria over here in the forest, and this is a kiwi plantation. And what they've done is they've built a barrier here to prevent the roots from here coming down through into the plantation, and that prevents the pathogen moving into horticultural situation. So you can establish physical barriers between them, prevent these roots getting in. You can use chemicals, but these things, so the soil fumigants may be only used by trained professionals, as not recommended in domestic situations. It may be able to be used in some gardens, but council gardens, but I would not be using it. If you're in this situation, you could easily just get a backhoe in and remove the trunk, remove the stump. There are some new fungicides that have had a direct impact on the pathogen, recently developed in orchards, and may have an application on shade trees in gardens. This is the cyproconazoles, the Alto, and I'll tell you how we've used that in a case study when I get down to it. Biological control. It's been shown that if you infect a stump with other pathogens, these are two infected stumps. You can see here the Armillaria fruiting on the infected stump here, but this one here has had Trametes actually inoculated into the actual trunk, and actually killed off the pathogen within the trunk. So Armillaria itself is not a good, it doesn't stay well in the soil by itself. It actually will get killed off by all the other fungi if you can promote it, other fungi to get into it. They have tried using what's called biological control using trichodermas, and this is mixed reviews in the literature. It's not really truly a biological control agent. It's more a biological pesticide. And so in my situations where I've used it for just to try and help kill off the pathogen in the soil after you remove the stumps of the actual malaria, remove most of the inoculum. So that's where these things, trichodermas, are actually used. You can try improving the tree health to resist it. Again, we use here. They've used this phosphonate again to inject into fruit trees in this case, and that seemed to give some protection from malaria. You also maintain good nutrition, and again, as with Phytophthora, it's the tissue water content of the actual tree that is an important part. And if a tree is overwatered, it tends to get so much water in it that armillaria tends to get going in the trees as well. In all cases, if you're going to use any fungicide to any plant for the first time, you always test for phytotoxicity. Hygiene, it's important to maintain good hygiene practices to ensure that all the root material is removed, and that should be bagged and securely removed and appropriately disposed of to ensure the contaminated material doesn't spread. Certainly don't use it as mulch in your garden, or do not shift the plants from an infested garden bed. This is another treatment that has been quite successful, is to expose the collars of the trees so that they dry them out. The pathogen doesn't seem to be able to get up into the trunk where it does the girdling if you actually expose the collars of the actual tree. This is an interesting little video that let's see if I can play it. Oops, now I've gone to go back. So as you can see from that video, that the drying of the actual collar region helped in the control of the pathogen. So just as a case study, Armillaria luteolina has been one of our major issues in the Museum of Victoria, where I don't know how many of you have been to the gallery, the Forest Gallery. It was developed and built in 2000, and large trees were transplanted from native forest from the Central Highlands. They brought in armillaria with them on their root systems as the transplanted tree. And so we've been working on that disease control since 2001. So, and we've developed the following protocols. You can see here, this is the Anothophagous cunninghamii that's rapidly died. And you can see the mycelium up underneath the bark of the actual tree. So this is the actual protocols. And if somebody would like them, I can print my notes out for somebody if you really would like them. I can actually leave that with Bill, and he can give you these notes if you want. But the main thing is plants removed at the earliest opportunity. You can drench the edges of the holes with the fungicide. It can fill the hole that's created by the removal of the trees with clean soil enhanced with a mushroom mulch, which helps to break down any remaining infected roots in the soil that you might have missed. Where possible, you leave the area fallow for at least 18 months before replanting. You can plant annuals on the top of it, but that's all. Expose and maintain the collars of any retrained trees, any trees that might be next to those ones that have died and been removed. You need to expose and maintain the collars without any soil or mulch up against it. Ensure that your watering is minimized and sprinklers not directed so as to not spray directly onto the trunks of the trees, and stopping high plant tissue moisture content. In the spring each year, drench or paint any exposed collars of any retained trees, infected trees, and adjacent tree subs with alto, and inject with phosphonate at the appropriate rates 2 weeks after an alto application. and then we can expose any mycelial fans in the bark at the collar of any retained infected trees. This involves peeling the bark back to healthy stem. And I'll show you this is sort of thing here. In June 2020, we had one of our, this is a blackwood in the forest gallery. You can see the massive armillaria present. So we peeled this bark back, we painted it with and treated it with alto, and this was in 2021. No sign of any fungus anymore, so it's actually been killed off within the tree, which is good because that was one of the last blackwoods they had in the actual gallery. So these are the other methods they can use. You gotta make sure your trees are well fertilized, that they you need to have a good monitoring program in late May, early June to find and locate any trees that have any armillaria fruiting bodies within the garden. You can then drench those around those fruiting bodies with a mixture of trichoderma and perhaps even alto if you need to. Any armillaria fruiting bodies you collect and you put in sealed bags and preferably heat treat them and then dispose with the general waste. That just prevents any potential for spores being blown onto any other wounds. And examine for any fruiting bodies from their source. So if you find it doesn't appear to be attached to a tree, but you get some armillaria fruiting body coming up, check down to see underneath the ground to see where it's coming from in terms of a bit of root material and remove that as well. So throughout the Dandenongs, we've got, this is where we know, for instance, this is Armillaria luteolina. It's a garden in Emerald. This is just this last June, which is when I looked at these particular plants, these trees. So we've got one at Emerald. We've got them in, this is down the Puffing Billy car park down in and the area where Bill and the team have been doing planting down in the Sherbrooke forest there. This is up at Ferny Creek. It's armillaria, and that was exposed when that windstorm went through and the trees that got blown over. And I found a friend of my place. I went up to help him out with some of his trees there, and found armillaria in the base of the trees that had fallen. And I did suggest to councils to have a good look at the blown trees to see whether they can take samples from them and then see how many of the trees that are blown over were actually infected with armillaria. There's ones here at Ferny Creek, in the native forest there, up near the kiosk there at Callista, and then these armillarias through here are not luteolina. There's one here at Monbulk. I believe that's in Bill's place, his own house. And then this armillaria here is a different armillaria. This is two actual Phytophthora locations. So the ones that we've had in the Sassafras Creek is this one here, Armillaria novaezelandia. You can see it's very different to the other armillaria. It's got this really shiny, wet look on it. It's not supposed to have, not to be as pathogenic. It's only supposed to be mildly pathogenic, if something else has actually caused the trees to be stressed, that's when it might become an issue. I've never seen armillaria here newly in the Dandenong Ranges, but I'm told that it's present, but it's apparently very rare. So Bill's work here, Bill and his team have checked all these stumps right through the Sassafras Creek. That's an amazing job that they've done, looking at all these trunks, and we've only found this Armillaria novaezelandia in these particular sites. So congratulations, Bill, and it's a great amount of work you've done. But and that's again a photo of an image of the various locations of the soil testing and the armillaria. So again, we haven't got any susceptible hosts. We looked at most of the work is done is in trying to remove the pathogen from the soil and trying to make sure that the soils are not too moist. You can stop watering, for instance. You can prevent mulch being built up around the trunks of the tree. That's a big way to improve the environment and therefore reduce the amount of disease. So what do you think? What do you think triggers a native root pathogen to kill a tree? There we go. One of the key factors appears to be following defoliation events. This one, for instance, the mountain ash psyllid, Cardiaspina bilobata, is one where you get a lot of defoliation of the trees. It particularly affects regnans, delegatensis, and Eucalyptus fastigata. This particular psyllid affects both juvenile and adult foliage in saplings and mature trees. But attack is cyclical and related to temperature, elevation, and slope. It caused extensive defoliation in the mountain ash forests in 1986, 1994, 2015 to 2018. In fact, it wasn't identified for the first time in 1986. It's first described. One of the other pathogens, target spot, Oligophyna eucalypti. This is another native disease of eucalypts. It causes a leaf spot and petiole lesion. So it gets not only on the leaves, but it gets on this petiole here. And what tends to happen is the plant, as soon as it gets infection in the petiole, the leaves tend to get shed by the tree. Again, in 1974, it caused extensive serious defoliation up here. You can see, I'll show you in the next slide, I'll show you Eucalyptus nitens in East Gippsland in 1974. And in association with the psyllid, it caused significant defoliation in E. regnans in the Central Highlands. So it seems to work in combination with the psyllid. So this is what the defoliation on the Erinaundra Plateau was like in 1974, all caused by Oligophyna. And when we looked at the areas in landscape images in 1994, you can see the extent of the defoliation due to the combination of Oligophyna and the psyllids throughout the areas. But the interesting thing was that in the gullies, while the defoliation was extensive across the sites, it was only the gully areas where you actually had tree deaths. And when I went up to these sites and got down in the gullies to try and ID the cause of them, the trees have only just finished defoliation, had a few leaves left on them, and yet the trunks of the trees were thoroughly infected already with secondary root pathogens. So it appears that the defoliation event has resulted in the trees becoming more susceptible to other secondary pathogens, and that's gone right up and killed the trees. Another defoliator, Mycosphaerellus, they're called Tetrasphaeria now. It's had its name changed recently. These are what we call tentacle crinkle leaf diseases. And you can see they could certainly have a big issue in plantations in particular, but also in native forests as well. Spasmodids were a big issue back in the 60s and 70s, and even more recently in 2001 in the Delegatensis up in the Upper Yarra, Upper Ovens catchment, and you get this sort of defoliation occurring across the plantations. And in this case, you do still get deaths in the gullies. Christmas tree, cross-mellor leaf beetles, they're another one that causes defoliations and may lead to wood decay fungi becoming an issue. Again, you can see the disease, you can see the leaf defoliations that occurred. More leaf beetles that are can cause defoliations occurring. Other psyllids, these other psyllids generally occurring in the red gum forests and mahogany gum down in East Gippsland. They cause defoliation of the whole of the trees. It's interesting that you can actually, from the air, you can actually pick the distribution of mahogany gum by the defoliation that occurs due to the Cardiaspina. Gumleaf skeletonizers, Uraba lugens, it's another one that causes significant issues in terms of defoliation. You can see why it's called gumleaf skeletonizers. Cup moth is another one that causes damage to eucalypts. And again, some of these are really pretty, but pretty to them, but they cause rather serious defoliation. And the last one I'll talk about defoliators is some of our vertebrates can also cause issues. I know we don't generally associate koalas with significant defoliation events, but in the Otway Ranges down near Grey River, a large population boom of koalas resulted in all of them, the trees being defoliated, and quite a lot of them died. And again, that happens with possums, can cause issues as well, and particularly in some of down the Mornington Peninsula. And you can see here where they put a band around the tree, the actual tree's recovering after preventing the possums from getting into it. So most of this time, it's our fault that these things are causing issues like this, because we tend to create these small patches of native forest that the animals can't move on, and therefore they stuck with the same trees all the time. And so we have to try and manage that as best we can. Wood borer is another one that you'll often see related to stress. When you get a stressed tree, these things actually can cause quite significant damage internally in the trees and cause them weaken them, and they can cause them to snap off or break off. How are we going for time? What time? The last one I'll quickly deal with is the bell miner associated dieback. This is one that doesn't occur much, fortunately, in the Dandenong Ranges, but certainly off towards Listerfield and a number of other places around there where you get these discrete patches of vegetation that we've created, that and the bell miners, and we prevent fire from getting into them because they're too close to communities, and we get understories, massive understory growth of weeds, and the bell miners love these things. They build their homes on the tops of these vegetation, and then proceed to farm psyllids on the eucalypts, and these psyllids then cause defoliation of the trees, and because the bell miners actually what they do is they chase away all the other birds that would normally feed on these things effectively, and the result in the populations of these things exploding. And again, the tree death tends to be via secondary root pathogens following the defoliation. It's not been caused by the psyllids, but it's caused by the defoliation leading to weakening of the trees. So I think I might leave that at that, and see how we go. Any questions?
Yeah, we do have a few, Ian.
Right.
I might paraphrase Sergey and Gay's questions. Actually, they're both commenting on Sassafras Creek, or asking what your thoughts are in that area, seeing as some of the mountain ash are looking pretty sparse. Do you think it's evidence of armillaria, or do you think it's a natural successional degradation of those ash?
Well, again, if you look at the ash species, when they start off, when it starts off, you're looking at seedlings coming up at 1,000 to 10,000 seedlings per hectare. When you get to a mature aged forest, it's down to 10 trees per hectare. Something takes them out over that particular time, and I think there's an association between armillaria and other root pathogens and defoliation events. I mean, there was a defoliation event in the early 2000s, and even in 2017, I think there was a defoliation event due to mountain ash psyllid, and those sort of things can weaken the trees and result in things like armillaria becoming an issue. I know we only found armillaria in I think 10 of the trees in the gully, but that's only because based on the fruiting bodies. I mean, other stumps may not have produced fruiting bodies, and there might be in the trunks of the trees that we couldn't ID. The only way to ID from the trunks would be to take mycelium and have them DNA tested, and that's a very expensive role. So armillaria may be playing a role in those things, and along with other wood decay fungi following other stress events that have occurred.
Yeah, great. And we've got a question from Matt, who's our local ranger in charge in the Dandenong Ranges, about whether armillaria has similar temperature constraints to Phytophthora. No,
it's much colder. It can actually occur in a much colder environment.
Yeah, and then there's one more question, which I know you've got an answer to. We had mountain ash come down in the storm in our creek at Alinda. It's pretty much mountain ash and black wattle regrowth forest. Would a more diverse replanting improve resistance?
armillaria can take out anything, frankly. Ash forests are primarily a single species forest with a wattle understory, and more. I mean, I'd love to put Myrtle Beach back into the Dandenong Ranges, but because it occurs in the Central Highlands and occurs in the Otways, it just seems to have missed the Dandenong Ranges. But though I think there is a successional situation in all of these things. I think what we get is you get a massive bushfire go through, the seedlings germinate, seed gets dropped into the gullies. The gullies are burnt out. The species that are in there get killed potentially, and then you get a massive regeneration occurring, and then over time these are reduced down, and then you get these drought and defoliation events where you've got these pathogens that are existing in the gullies. They're hiding in there because that's the wet areas. Then you get those trees dying off, and the rainforest species taking back over again. So they're having this little battle between themselves, who's going to occupy the gully. But I think that's part of successional parts of our native forest. It might be a 300 or 400 year battle, but unfortunately we've got a situation now with climate change, which we might be getting warmer conditions, which is something that concerns me. We were getting things like the mountain ash psyllid. We didn't know of it before 1986. Where have they suddenly turned up from? They should have been around it. These warming up conditions are resulting in these things becoming more of an issue. I'm concerned with things like myrtle rust, which I didn't get a chance to talk to you tonight. Myrtle rust, which is an introduced pathogen to Australia, and it's spreading down the coast, and it's got into Melbourne now. It hasn't got up into the Central Highlands, but we know ash species are susceptible, but it's, I think, generally considered to be too cold. But what happens as it gets warms up is myrtle rust going to start taking out seedlings. And there are a lot of other pathogens. I say we can get a chance to talk about Phytophthora ramorum, which is one that we really need to be careful of, because it's one that's a cold temperature Phytophthora. It quite happily lives at 0 degrees, and its optimum temperatures at 15. It loves Nothofagus, it loves regnans, it loves delegatensis. Because I had a student, when I was first heard about ramorum, I organised a student to go across to the States and have a look at because we have native species in gardens in the States, and they've got mountain ash growing in some of the botanical gardens, and we managed to get hold of some of that material and test Phytophthora ramorum against mountain ash, and it was an incredibly sensitive to the pathogen. So I'm really concerned if that ever gets here, that we could have a big issue there.
Yeah, one to watch out for.
Mm.
Well, I think that's closed off our questions. We've covered most of them. You did actually answer some of them just through your talk as we went, so that was really good. But thank you, Ian. That was fascinating. I learned a huge amount, and I'll certainly be coming back to you with a lot more of my own questions when we've got more time. But Helen, I'll hand back to you, and you can sort of round us out.
Thanks, Alex. Thank you so much, Ian. I'd like to echo Alex's comments. That was absolutely so informative. I learned so much, and I'm sure everybody did. And I think we can now have much greater understanding of this in our forests. And when we're seeing some of these fungi, we know what we're looking at. And so, yeah, it was very, very useful. And I'd like to thank you very, very much, Ian, for your time.
That's right.
For sharing your knowledge and your expertise. It was really excellent.
I hope you got the concept of the disease triangle, in the sense that one thing on its own is not necessarily the cause. You might have more than one thing. And on its own, like the pathogen may not cause an issue, but it's only when you get something else appearing that defoliates, that causes an impact on the host, that then the armillaria might become an issue. So it's what causes the death is a combination of them rather than one thing on its own. Eucalypts tend to withstand defoliation quite well, except in situations where you've got wood decay or armillaria secondary pathogens in the gullies. So I'm just trying to answer one of the questions. That's all.
So yeah, thank you very much. We, as you know, we have recorded this, so Bill will be your conduit, and we will send you out the link so that if you want to have a look at it again, you can, or if you'd like to pass it on to someone who might be interested, you're most welcome to do that. So thank you, Ian, for allowing us to record your session.
Yeah.
Yeah. And thanks, Bill, for organising all of this, and hope we see you again. There will be more quad events like this, so hopefully we'll all catch you again in the future, and hopefully one day face to face soon.
Yeah, it'd be nice.
Yeah. So thank you again, Ian.
Pleasure.
Thank you.
Thank you.
Bye, everyone.
Bye.
Bye.